Okay, so we were talking about this transfer of light energy into chemical energy and then moving up through what are called trophic levels. So as you move from producers to consumers and then the consumers to whoever's consuming them, you're moving up in what are called trophic levels. And so you can also have that energy from primary producers go down into decomposer communities. And so decomposer food webs are a little different than primary producer food webs. And oftentimes, decomposer food webs tend to be longer and more complicated. So this figure here is showing you on the left, live green maple leaves, and then crickets that eat them, and robins that eat crickets, and hawks that eat robins, right? So you can see there are 4 trophic levels in that green food chain. Now, if you have dead maple leaves, then you might have bacteria eating the leaves, and then earthworms eating the bacteria, and then the robin eating the earthworm, and then the hawk eating the robin. So you have actually an additional level in your trophic in the brown or the decomposer food chain. And then this is a terrestrial food chain, but what's really interesting is when we move into marine systems, marine food webs are much more complex than terrestrial food webs. And this has some implications for some stable isotope chemistry that's really fun and cool. And hopefully we'll get to talk about. So you can visualize food webs a little bit more complexly when you're not thinking about just that simple chain, because of course robins can eat both crickets and earthworms, right? They're not necessarily completely omnivorous, but they're more generalist, so they eat across multiple organisms. And so here you can see that energy flowing from various kind of basal sources, from dead animals, dead leaves, live leaves, and rotting logs, into lots of different types of primary consumers, and then up into secondary and tertiary consumers. In reality, food webs look more like this, though, right? Like there's so many species in ecosystems, and there's so many interactions happening that to really get a good picture of the food web requires this very complicated network analysis. But this is more advanced. This gets very advanced very quickly. So we're really just going to keep things simple, thinking about these relatively simple food webs. Okay, so for example, this is partly why marine systems are more complicated, is that in a typical forest, you have live leaves and only about 5 to 7% of the live leaves go to a primary consumer. And then most of the leaves enter the detrital or the decomposer food web, and about 93 to 95% end up feeding decomposer food webs. Now that's different in a typical marine system where live algae tend to support more like 35 to 40% of the primary consumers, and the dead algae tend to support less, but still a majority go to primary decomposers. So this is one of the slight differences between forests and oceans. And we'll talk about others too. So another way to think about food webs is in this kind of idea of a pyramid. And so here's an example. If you have a whole lot of plants, so pretend you have 1,000 plants in your system, 1,000 plants, then a small amount of those plants gets eaten by primary consumers like crickets. And so you can only support about 200 crickets from 1,000 plants. About 80% of the energy gets used and kind of released, and only about 20%, 10 to 20% of the energy gets transferred from one trophic level to the next. And so you can see we start off with a large amount of green stuff. We can produce, a pretty good number of crickets. But then again, there's this loss of energy. It's called there's a low efficiency of energy transfer from one of these levels to the next. And so you can produce 200 crickets, but those 200 crickets can only produce 30 robins. And then the 30 robins can only support, say, 3 hawks at the top. And so we have this kind of transfer of only about 10% of energy from one trophic level to the next, which is why you see very few large predators on the landscape, right? There's just not enough energy to support them. This is also similar if we have a toxin in the environment, and some toxins are held in our bodies, especially those toxins that are stored in our fats. And so things like DDT and um, um, persistent organic compounds, they show up in our ecosystems and primary producers in pretty low rates. But then the organisms that eat those plants accumulate all of the toxins that were in the plants themselves. And then the toxins are in higher concentrations in the primary consumers. And the secondary consumers that eat those primary consumers continue to magnify the toxins until you get to tertiary consumers where the toxin loads can be ridiculously high. Okay, so the biomagnification of toxins can be a real problem for top predators in particular. And this was true for DDT and other pesticides. Here you can see some really odd advertisements from quite a long time ago now, but DDT was really pushed as a pro thing, basically like something that you'd really want in your house. They made children's wallpaper that was impregnated with DDT to protect them from mosquitoes. And exposure to DDT, which was used to control mosquitoes, and other things like dioxins in the environment, which was a waste product from industrial uses, resulted in the egg failures of many top bird predators, things like eagles and hawks and peregrine falcons. And the book Silent Spring by Rachel Carson tells this story and really brought this to, highlighted it for us so that we could understand DDT. And in fact, the beautiful thing is that many bird populations have actually rebounded from these very low populations since the 1970s. And evergreen students in 1974 and 1975 were at the forefront of DDT research with faculty members Steve Herman and Mike Bugh. So there's a long history too with evergreen. Here you can see a DDT truck at the beach with kids running behind it because it was fun to run in the fog. And you can see pictures of eggs that were crushed by their mothers while they were trying to incubate them during periods when the bird shells were thin because of DDT. So this is a picture from the New York Times front page story. And I would say Rachel Carson was demonized a little bit, definitely by industry, and but her perseverance in the face of that attack was important for moving forward with protecting bird populations in particular. Unfortunately, DDT is still used in lots of other countries around the world, and so we're not completely out of the woods when it comes to this kind of pollutant. And the last thing we're going to talk about in terms of trophic structure is this really cool case study with wolves. So here's an example. You can see grasses, aspen, cottonwood, and willows. So grasses tend to be fed on by mice, and then mice are preyed upon by coyotes and hawks. But the other three, the aspen, cottonwood, and willow, are all related to one another in the family Salicaceae, and they are preyed upon by beaver, elk, and mule deer. And the beaver are preyed upon by coyote, but wolf prey on the elk and mule deer. And so this kind of interesting interaction among these organisms led to some really interesting changes in ecosystems. So here's a picture showing what Yellowstone looked like without wolves on the left. So when wolves were not yet reintroduced, we have elk down by the river, not very much riparian vegetation, no young aspens, and just a few coyotes. And then when you add wolves back to the system, that pushes the elk away from the riverside. We get recruitment of young aspens, nice riparian development, and a whole bunch more diversity. So this is an example of kind of a trophic cascade. And we have this shown more close to home here on the Hoh River, where we actually saw that elk would push down black cottonwood populations. And so when elves were extirpated, we had more elk foraging in riparian areas, decrease of riparian plants, and it changed the way that rivers moved through the landscape. So that's kind of an interesting tag along to trophic dynamics. Okay, take another 10-minute break and we'll come back for the last lecture.